I'm going to introduce Jimmy Yee. He has a lab at UCSF that's interested in understanding the molecular basis of natural human variation. For the past few years, they have developed new methods to perturb, measure, and model genome function, and have applied these methods to study how genetic variation interact with environmental perturbations to create the diversity of immune responses observed in both healthy humans and patients with autoimmunity. So, Jimmy?
All right, great. All right, thank you, Giovanna, for that introduction, and 10X for having me here, and all of you for sticking around. So I'm going to tell you a little bit about the efforts ongoing in my lab for the past 2 years or so to try to build a molecular survey of gene expression in primary human immune cells across a large population cohort. So I'm going to give a brief introduction on why we want to do this thing, why do we want to build the human immune census. And so here's an experiment that many of us have performed, where we're looking at gene expression differences in bulk between two conditions. This could be between healthy individuals versus diseased individuals, responders versus non-responders, and for this audience, looking at the effects of genetic variants on gene expression. As Molly introduced, this is often called expression concentrated locus. For simplicity, I've only indicated 2 genotypes. So we do these experiments in bulk, and what could be happening at the level of a single cell is that that difference in gene expression could be explained either by a change in cell type proportion, or a change in cell state, or maybe a combination of both. And as a postdoc, we really spent a lot of time to try to figure out that second case, which is, are there genetic variants that would have context-specific effects, that is, in different cell types or in response to different activations, you would observe a genetic effect, but in others you won't. And so this led to a number of papers we published in the past couple years, looking at peripheral blood mononuclear cells from hundreds of healthy donors. And we did basically 3 arms of the ImmVar study. The first arm is comparing genetic effects on gene expression in monocytes versus lymphocytes. So monocytes are the innate immune sensors in our blood, and lymphocytes are one arm of the adaptive immunity. And so we first did this with microarrays. And then we also did a similar experiment looking at the response of monocytes to in vitro activation, as well as the response of T cells to in vitro stimulation. And we did this initially with nanostring because of the number of experiments we had to do. And then we recently published on using RNA sequencing as well as ATAC-seq to look at changes in not just overall gene abundance, but in chromatin state as well as RNA structure in both dendritic cells and T cells. So I'm not going to talk about these results too much, but to summarize that, basically what we've observed is that genetic effects on gene expression is highly context-specific across cell types as well as different stimuli. And then what really motivated what I'm going to talk about next is that these experiments were incredibly expensive and laborious and biased. We were only basically looking at 2 cell types in blood. And there's no estimate of cell type proportion in any of these. And so what we've been wanting to do is build this thing that we're calling the human immune census. What is it currently? Well, sort of this first pass version of it is single-cell RNA sequencing of about 1 million peripheral blood mononuclear cells. But instead of doing this experiment over 1 or 2 individuals, we're doing the sequencing over 200 samples, about 5,000 cells each. Initially, we're looking at differences between lupus patients as well as healthy controls. Our goals are to quantify cellular and molecular variation between individuals. In this particular study, we're interested in identifying biomarkers that's predictive of disease status and severity. And finally, to associate context-specific gene expression variability with genetic variation. And so, many of you are probably familiar with this. What we've been using in the lab is the 10x Genomics 3' single-cell RNA sequencing solution. And on the right-hand side is my cartoon representation of the technology. Basically, you have an oil emulsion that contains both a labeled barcoded hydrogel as well as a single cell. And for each one of these assays, you can sequence anywhere from 1,000 to 10,000 cells per channel. At doublet rates that are between 1% to 10%, and the doublet rate increases linearly as the number of cells that you want to sequence. So as a thought exercise, suppose that we want to build an immune cell census over 100 donors. We want to get 1,000 cells per donor. One way to perform this experiment is by loading one sample per well, targeting 1,000 cells. But I said earlier, per channel, you can actually sequence up to 10,000 cells. So alternative strategy would be to load cells from 10 people and target 10,000 cells in each well. So obviously, if you do this, we increase the sample throughput, and we can also eliminate some of the confounding effects from perhaps manufacturing or just handling lots of different samples. But the obvious caveat and the downside to this experiment would be that we would increase the doublet rates, and we would also not be able to identify sample identity. And so then how do we construct the human immune census at scale? And what we published now last year is the development of a simple experimental workflow called Mux-seq and an algorithm called the Mux-seq that enables population-scale single-cell RNA sequencing. And the way it works experimentally is very simple. It's exactly what I just said, which is we're going to try to mix cells from different people together. They're genetically different. And we're going to flow it into one 10x channel. So instead of the cells having the same color, now each cell has a different color. And within a droplet, in addition to being labeled with a synthetic cellular barcode, there's 750,000 of them, each cell is also labeled by a natural barcode, which is genetic variants that are occurring near the 3' end of a transcript. And the goal of our algorithm, the Mux-seq, is to harness naturally occurring genetic variants. These are single nucleotide polymorphisms, to demultiplex, which means to assign each cell correctly to the donor of origin, and to identify cases where you have doublets. So I'll just walk you through sort of an example of how this actually works. It turns out that capturing a natural genetic barcoding single cells is lossy. Here is just a representation of the genetic barcode of an individual, and this is all going to be coding sequence near the 3' end. And if you look at the transcriptome of a single cell, because of shallow sequencing, you're probably going to capture only a fraction of the natural genetic barcode. And in most cases, you will not have the resolution to resolve a heterozygote site versus a homozygous site. Now, furthermore, if you have two cells that came from the same person, and you just compare the genotype from these two cells, you might not be able to tell that they came from the same sample if you didn't already have the barcodes initially. But it turns out that if I gave you that genetic barcode of an individual, and you can obtain that using cheap genotyping or exome sequencing or even bulk RNA sequencing, you can actually